Good morning, everyone. I am Aravinda Chakravarti. I'm a professor of human genetics here at NYU School of Medicine. And I've had interest in this particular subject for a very long time. We are the committee assembled by the National Academies to study the use of race, ethnicity, and ancestry as population descriptors in genomics research. As you will see, we will come back to this topic again and again because I want everyone in the audience to recognize that we're talking about something quite specific. First, I want to welcome my fellow committee members, as well as the speakers who graciously are going to give of their time today. And I want to acknowledge the audience that's listening in on this webcast. I've introduced myself and now really have the pleasure of yielding to Charmaine Royal, who is the co-chair of this committee. Charmaine?
Thank you, Aravinda. Good morning, good afternoon, good evening, wherever you are joining us. I am Charmaine Royal. I am professor at Duke University and like Aravinda, have been studying issues of genetics, in particular, the issues at the intersection of race and genetics for a while. And it's my honor and privilege to be a co-chair of this. Welcome to all of you who are joining us, and of course our speakers and our committee, and looking forward to the conversations today. Thank you.
Thank you, Charmaine. So this study has been sponsored by the National Human Genome Research Institute, but they are not alone. There are 14 other NIH institutes and centers that have also sponsored the study and are eagerly looking forward to the report. I want to specifically thank the speakers for taking the time to speak to the committee. But before we proceed and I hand it over to the moderator, I'm going to read a few disclaimers for these public sessions. So as a co-chair of this committee that I told you about, first, I want to welcome everyone to the first open session of this committee's second meeting. I want everyone to note that this is an open, on the record, information gathering session. The committee is in the process of hearing from a number of experts, and we are in the process of assembling the many voices and assembling published material and other materials so that we can examine and discuss this topic in the course of making our findings, conclusions, and then recommendations. I want to remind everybody that we should all remember that we as a committee have made no conclusions, and that it would be a mistake for anyone to leave here today thinking that it is otherwise. The comments made by individuals, including members of the committee today, should not be interpreted as positions taken by the committee or of the academies. These are our individual views that you may hear today in our questions. I've already spoken about the sponsorship of this study by 14 institutes and centers in the NIH. And if you want to know the specific centers and institutes and offices, then please go and look at the study website where they're all listed. So for those of you who may not be familiar with the National Academies committee process, this is the second of 5 planned committee meetings. Some of these meetings, such as the one today, will include public sessions where we will all have the opportunity to hear from many experts in the field and receive public testimony. Please remember that the committee will discuss this and other materials before we even begin writing the draft report. Moreover, once the draft report is prepared somewhere along later this year, it will undergo a rigorous review by experts who are completely anonymous to us in the committee. The committee then has to respond to these questions and the critiques, and we have to make appropriate revisions to adequately satisfy the Academy's report review committee and the chair of the National Research Council before it can be considered to be an official NRC report. We expect that the report will be released in early 2023. For further information on the study, including future meetings and the full statement of task, as well as these disclaimers, you can find them on the project's website, which I would refer you to look at regular intervals to see the progress. So what's the purpose of this committee? We are being asked to review and assess the existing methods, benefits, and challenges in the use of race and ethnicity and other population descriptors in genomics research. I want to remind all of you that in scientific studies, one of its major features has to be consistency and replicability. When any one of us or groups of investigators design a study, we have to describe the human subjects that we use so that these findings can be tested by others, replicated, and extended. And that's the specific context in which this open session is being held and this committee is working. Our, of course, final aim is to develop feasible and logical approaches to advance the appropriate use of descriptors of both populations and individuals so that they can appear in published genomic research studies with the indications of replicability and extendability that I spoke about. So the open session today provides an opportunity for us, the committee, to hear from outside experts, because this will inform our internal discussions and writing. In the interest of time, it's unfortunate I cannot introduce to you all of our committee members, but there are 17 of us, and the full roster and the biographies are available on the study website. So to recap, today we have 3 sessions. In the first session, we will be looking back as to how population descriptors have been used in genomics research historically and how they're used today. In the second session, we will take a forward look and ask our presenters to give us their views and their considered thinking on how population descriptors may be used in a much better fashion in the future. After the speakers have provided their remarks, we will then have time for discussion with the committee. The third and final session of today's workshop is based upon the committee's call for public comments. We have asked for input on this subject from a variety of stakeholders and a variety of individuals interested in this area. And we've asked a selection of the many commentators who provided questions to join us here today. It's unfortunate we couldn't accommodate any more of you. To those listening to the webinar today, I encourage you on behalf of my colleagues to submit a comment if you have not done so already. And with that, it is my great pleasure to introduce our fellow committee member, Dr. Sandra Lee, who's Chief of the Division of Medical Humanities and Ethics at Columbia University. Sandra?
Great, thank you, Aravinda, for the introduction. I'd like to extend my welcome to our public audience and the organizers of this workshop and to our speakers today who have generously devoted time to be with us. It is my privilege to be moderating our opening session today on the historical and current uses of population descriptors. I see this as a critically important discussion that will provide the socio-historical context for how categories, labels, descriptors have been attributed to human beings that will help us understand current taxonomies, what they are, how they were created, and for what purposes. We are fortunate to have scholars and scientists who have rich expertise to guide our understanding of the use of population descriptors in genomic research and to share conceptual tools and a socio-historical framework for the committee's work. Before we turn to the panel presentations, I do have some housekeeping notes. We'll have 15 minutes presentations from each of our speakers. This will be followed by a discussion with the committee members. And at that time, we'll also be taking questions from the audience who have joined us by webinar. To our audience, we welcome you to submit your questions in the Slido box underneath the webinar broadcast. And you can do this at any time. We will be keeping track of those that come in and referring to them after the committee discussion. So for the sake of time, I will not be reading the speakers' full biographies, but you may find those in the briefing book in the meeting materials section of the event's webpage. Our speakers have been chosen for their expertise in specific scholarly and scientific work that bears directly on the questions that we will take up in this session, and I encourage you to refer to their bios. Please note that we will only take 1 or 2 clarifying questions from the committee members immediately following each presentation as time permits. So I ask that you please hold other questions to our time after the presenters have spoken, when we will engage in what I hope will be a robust discussion. So today we'll first hear from Dr. Pilar Osorio from University of Wisconsin-Madison, followed by Dr. Joseph Grays from North Carolina A&T State University, then Dr. Andrew Clark of Cornell University, and finally Dr. Rena Bliss from Rutgers University. So with that, our first speaker today is Dr. Pilar Osorio, who is Professor of Law and Bioethics at the University of Wisconsin-Madison Law School and Medical School. Thank you, Pilar.
Thank you, Sandra. I am unable to turn on my video, so somebody else, here we go. There. Now I can turn on my video. Thank you, everybody. So I am the lead-off. I'm the lead-off batter here. And I am not going to be using slides. I might be the only person who is not. And so first of all, I want to thank the Academy and the committee for inviting me to be part of this really interesting day today. And I'm going to start with a little bit of background, in part to maybe situate this discussion for the broader audience. So our panel is supposed to focus on the past and present with respect to the use of population descriptors and genomics research. And I'm not a historian, but I have been involved in various ways with human genomics since the very beginning of the Human Genome Project. And early on, I became associated with the Program on Ethical, Legal, and Social Implications of Genomics, that is referred to as ELSI. So I became associated first as a bench scientist, a molecular biologist who had a deep interest in ELSI. And later I became a legal scholar and ELSI scholar. And I've been part of several large genomics projects, such as the 1000 Genome Project and the Human Microbiome Project. I've also written on the use of concepts of race and ethnicity in genome science, and on how the concept of a population is used in genome science. So today I'll speak from my experience and scholarship, but I'm not a historian. I don't even pretend to be one on TV or in presentations like this. And the first thing I want to say is that the questions addressed by this committee are not new. What counts as a population in genome science and how to describe populations has been a widely discussed and somewhat fraught topic from the beginning of the Human Genome Project and what is now the Human Genome Institute. The National Institutes of Health, many genome scientists, and even Congress were worried at the outset of the Human Genome Project that the processes and results of human genome studies might be used to support racist or white supremacist or even ableist views in the world. And given the history of eugenics in our country and our world, this wasn't a theoretical possibility, but one with historical precedent. So most of the concern about population descriptors has been on the use of race and ethnicity labels and how those concepts do or don't relate to genetic ancestry. So as I say, this conversation has been going on for years. I and many of the speakers on this for this day and on this panel have been part of those conversations for upwards of 2 decades. And despite the fact that these conversations have been going on for years, the very existence of this committee suggests that we really haven't reached any resolution. And I think perhaps this has come up as a topic for a National Academy committee, in part because of a desire for large genome data sharing repositories to have methods of standardizing population descriptors. But if this conversation's been going on for a long time without much resolution, that suggests that perhaps we're asking the wrong question, or that there are some important structural barriers in the practices of science that have prevented us from making progress. So I'm going to talk about that in a minute, but I want to start by saying I think we may also have ignored to some degree a predicate question, because we've tend to focus on the use of race and ethnicity labels and concepts, but we haven't had as much discussion, and I hope this committee will have discussion on how we understand the notion of population and a population. And I think in general, what I see in the literature is that genome scientists want some definition or notion of a population that they think is genetically meaningful, both to drive sampling for genome studies and analyses. And by genetically meaningful, we probably mean something related to the history of human migrations to different parts of the world, or historically restricted periods where some group was mating more within a group than outside of it. And so geneticists, for instance, have long resisted proposals to do something like just grid sampling in a city or a county or a region of the world. And in part, I think they have resisted that kind of proposal. Those proposals were perhaps never very serious, but also they were never very serious, and they've been resisted because genome scientists don't think that's a meaningful way to understand human beings. And they might or might not be wrong about that, but it's not very consistent with ideas about a population that have long tradition in population genetics. But I think there has been some work. Well, I guess I would just say that the term population is actually not exactly a term of art, not even in genome science. And just as an example, which is not a genome science example, I'm working on a project now where it's common for scientists to refer to, quote, the population of pregnant women. Now, this is not a notion of population that reflects some, history of migrations of peoples around the world. It's a temporary grouping where people have no commonality except for the fact that they are pregnant. But that terminology and that conception of a population of pregnant women, it's useful for certain types of science, even if it doesn't reflect something about history of population migrations. I think that work by Fujimura, Rajagopalan, and others suggests that even among genome scientists, the term population is used in ways that sometimes elide different concepts for grouping peoples. So genome scientists don't even always use the term population the way population biologists might think it should be used. And the problem with that kind of linguistic slippage is that it can also be conceptual slippage. And in particular, it can make social categories, such as categories related to race and ethnicity, appear more related to population migrations and sort of genetic similarity than they actually are. It can downplay the ways in which those categories are social and are constructed and have implications because of social hierarchies. So I bring this up now because I think a committee on population descriptors ought to say something about the term population and not just about the term about those descriptors that have to do with race and ethnicity. So as I said, the conversation about uses of language relating to race and ethnicity is an ongoing one in this community. And nonetheless, studies have shown that if you look at published literature, that race and ethnicity labels are used in a multiplicity of ways, that these are often inconsistent from study to study. And this continues despite many meetings, working groups, and studies that attempt to clarify the meaning of these descriptors. So as I said earlier, perhaps we're asking the wrong questions in all of these meetings, or perhaps there are significant structural impediments to making changes, or maybe both of these things are true. So in 2005, I published a paper with Dr. Troy Duster where we argued that basically we argued against this notion of trying to achieve consistency. And we also argued against this kind of discussion that has been going on in the scientific literature and in a lot of meetings where the questions about race and ethnicity descriptors are being asked as either should we use these or should we not? And our point was that we thought the important question was not should we use them or should we not, but when, where, why, and how should we be using these descriptors? And I think another question is, even when these might not be the best ways to understand the group of people who you are studying and how their groupings might be relevant for answering your scientific question, why is it that people tend to snap back to a few very simple racial and ethnic categories, particularly scientists in the US? And so I'm going to come back to this point about when, where, how, and why as really disrupting the notion of standardizing. But I do want to say that in my own work, I found that the reason that people snap back to like a very simple set of race and ethnicity descriptors is not necessarily because they think that those descriptors are the most scientifically relevant way of describing research participants who are in their data sets. So one thing that I've heard from many scientists is that if I don't use these descriptors, my work will not be cited as often as I think it should be or could be because it won't be viewed as comparable to previous or future studies. And so I think it's very clear, and even in some of Aravinda's opening remarks, that part of the goal that people have in trying to standardize race and ethnicity descriptors is to create a sense of comparability across time and across studies to promote reproducibility and the validity of the science that we're doing. And I actually think some studies that I haven't seen and would be worth seeing are studies just asking scientists why they chose the population descriptors that they chose, or experiments labeling the same data set slightly different ways and asking genome scientists how likely they are to cite that data or use that data, so that we could test some of these assumptions that scientists have, that if they don't use a certain pretty simple-minded set of race and ethnicity descriptors, that could sort of impede it could decrease the impact of their science, right? Another approach that has, I think, more recently been taken is to sort of expand the number of categories for race and ethnicity that people can use to describe themselves, right? And so this is an approach taken by the All of Us study, which has developed a set of population descriptors that is much more detailed and elaborated than other similar categorizations, right? So for instance, they have a number of categories that allow people to describe their Middle Eastern ancestry, or the idea that they come from a family that migrated from a Middle Eastern country. So that definitely captures important aspects of identity that have been missed in other population labeling that we use for research. But I think it still doesn't get at the point that these labels matter only they are meaningful in a particular time and place, right? And what it means to be South Asian or of Middle Eastern ancestry, it's very different in today's world than it was, pre 9/11. It depends on whether you live in, Georgia or New York City or San Francisco or Los Angeles, in terms of the access to resources that somebody has, in terms of the experiences of racism that they might have. So even as we might add more population descriptors and we might actually be more respectful to participants, this doesn't mean that we are capturing something that is standardized even within one historical or one time period. Let me put it that way. And if we go across historical time periods, even very recent history, again, the meaning of these categories varies. And so even if we standardize how we measure or how we ascertain race and ethnicity, that doesn't mean that we're going to be gathering data that are comparable across time or place. And one of my concerns is that we fool ourselves into thinking that there should be a comparability across time and place by creating some very elaborate set of recommendations on how to standardize the use of race and ethnicity descriptors. And just to give a little more specificity to this problem, I go back to something that, a question that came from a pharmaceutical company research director 15 years ago when I was giving a talk at a major conference. And this person came up to me and said, we really have a problem because we're running a big national multi-site study of a drug intervention. But our site monitors have realized that a person who self-identifies as Black in our study, at our study site on the East Coast, might self-identify as white or Latino at our study site on the West Coast. And is this a problem for our study? And I love that question so much. And I still love that question. And I go back to it all the time because I think the answer, of course, is it depends. It depends on whether this makes your study more complicated, but it is the reality of the world that even in a particular year, what it means to be Black or Latino or white depends very much on where you are, even in the United States. And when you start talking about comparability across data sets from different countries, this question becomes even more complicated, right? Because how people understand who is Black or not Black in Brazil is very different from how they or in Mexico, it's very different from how we understand it here in the United States. And I also just want to add that I think describing populations is something, it arises both during the primary data collection and generation and also in secondary data use. And part of what's driving this conversation now is a question about secondary data use and how we use data, aggregate data from different studies. And, so I have personal experience. Anybody who is part of the 1,000 Genomes Project has experience of having written a paper where we talked a lot about how we described the particular populations, the particular groups of people we were sampling, how they wanted to be, what descriptors they wanted attached to their samples, and why it wasn't, for instance, appropriate to generalize from samples collected in one community from people identifying as members of one ethnicity in Nigeria. It was not appropriate to generalize that to all people of Africa or all Black people. Nonetheless, I saw many papers published using those 1,000 genomes data where people explicitly just generalized from that one small sample set to and referred to them as the African samples and the data as the African data, right? So I think regardless of how careful we are with our descriptors in the initial data generation, that doesn't mean that everybody is going to be similarly careful.
Laura, we have about 30 seconds or so to wrap up.
Great, because I'm just at the end. And I just want to say that I think standardization of population descriptors is a misguided goal, that it creates a false sense of comparability across data sets. And it might actually also be disrespectful to participants. And instead of standardization of population descriptors, I think we ought to spend much more time in our genome science collecting data about culture, social experience, social status, and environmental exposures. And I think if we had good measures of those things, we're much more likely to have replicable genome science. All right, I will stop there.
Great. Thank you so much, Pilar. That was a great opening to our session. So our next speaker is Dr. Joseph Graves. Dr. Graves is a professor of biological sciences and associate director of the Triangle Center for Evolutionary Medicine. He is based in the Department of Biology at North Carolina A&T State University. Joe, I'll hand it over to you.
Thank you so much, Dr. Lee. So I will also thank the committee and the Academies for holding this very valuable and necessary discussion. By training, I am an evolutionary geneticist, and now you would call me an evolutionary genomicist. I have done my work in fruit flies and in bacteria, never actually done genomic studies of humans directly. Neither am I a historian or a philosopher of science, but I am definitely well into the literature of history and philosophy of science associated with biological and social conceptions of race. I've written 3 books on this subject. The first, I began to work on this in the late 1990s, The Emperor's New Clothes, Biological Theories of Race at the Millennium. The second, a few years later, The Race Myth, Why We Pretend Race Exists in America, which was published first in 2004. And just last December with my colleague, Alan Goodman, who is an anthropologist, Racism, Not Race, Answers to Frequently Asked Questions. And honestly, after I wrote The Race Myth, I hoped I would never have to write another book about race again, but here we are. Now, the things that I say in this talk are also based upon other publications that I have focused on this question of race and biological and social definitions, which are listed here. And I believe we're going to give our slides to the committee, so anyone who wants to take a look at these will be able to have access to them. Now, over the last couple of decades, I have often heard people say that race is not a scientific concept. I've even heard people say, or quote me as saying that. So this notion has become a platitude amongst many people in the humanities and social sciences. But this idea fails because it conflates 2 very different concepts. First, the biological race concept, which has developed over the last couple of 100 years, beginning first with descriptions of morphology or phenotypes, and then in the 19th century being focused more on geographical location. Then in the 20th century, after the neo-Darwinian synthesis, shifting to discussions of population, particularly the frequency of genes within and between populations. And finally, in the latter portion of the 20th century, focusing on the idea of unique evolutionary lineages within species. Now, one of the points I want to make about the biological race concept is that it has never been only driven by human biological variation. The biological race concept has been deployed to understand variation in a wide variety of species, of which humans are just one. On the other hand, the socially constructed or socially defined race concept arbitrarily utilizes aspects of morphology, geography, culture, language, religion, or pretty much any other thing someone can think of, but it only does so in the service of a social dominance hierarchy. So when Alan and I wrote Racism, Not Race, we wanted to make it very clear that it was racism that had created the socially defined groups within the human species, not an objective examination of biological variation within our species. That was never the case. It was always social contradictions of the way humans were living, particularly after the age of European discovery, along with colonization and wars of genocide, that these concepts were invented and came into widespread use. Now, there's a growing recognition of the social construction or social definition of race now in biomedical research. For example, Merrill's 2017 Introduction to Epidemiology states that race is a socially constructed variable based upon the idea that some human populations are distinct from others according to external characteristics or places of origin. Now, note that they don't say that these differences in the prevalence of disease are caused by genomic differences between these populations. Now, to go back in time, we need to think about how and when biologists and naturalists began to think about variation. And so I can arbitrarily start this discussion with Carolinus Linnaeus, who in his 1735 Systema Naturae began to talk about variation in a variety of species. Now, it is notable that he does not talk about humans until the 10th edition of this work, which was published in 1758. Here he described 4 subspecies of humans: Homo sapiens americanus, Homo sapiens europaeus, Homo sapiens asiaticus, and Homo sapiens afro. Now, Linnaeus did not explain how or why these groups were different. He relied on morphological and behavioral features. For example, Europeans were described as gentle and optimistic and inventive, while Asians were described as stiff and greedy. Now, Linnaeus never left Europe, so he based his judgments on information he received from travelers' reports. And of course, these individuals were far from being objective in their observations. Now, jumping forward into the 19th century, these individuals I call the Four Horsemen of American polygeny. And one of the points that I make in The Emperor's New Clothes, and one that's not widely recognized by modern scientists who have less appreciation for the history of biology than I think they should, is that biology up until the end of the 19th century was primarily driven by special creationist ideas, particularly from Christian religion. And so one of those ideas was that of the separate creation of the so-called races as in fact different species. Now, the major figures in this were people like Samuel Morton, Josiah Nott, George R. Glidden, and Louis Agassiz. All of them advocated some form of pre-Adamism, which meant the separate creation of Adam and Eve for the different so-called races, and that in fact Agassiz connected these different species of humans with specific zones of creation, each of which had its own flora and fauna, as well as its own species of humans. Now, Morton was one of the people who provided evidence through craniometry that these groups were in fact different. Josiah Nott was a South Carolina physician. George R. Glidden was the son of an English businessman, and they were the chief popularizers of polygyny in the 19th century, particularly in their works Types of Mankind, which was published in 1854, and Indigenous Races of the Earth, which was published in 1857. Nott, of course, as a South Carolina physician and a proponent of slavery, was acclaimed throughout the South for his lectures on, quote, niggerology. Now, at the same time, Charles Darwin was also interested in the question of variation within species. And he was interested in this question in association with his idea that the origin of new species was the result of natural selection. Now, he didn't talk about humans in the origin of species for a very good reason. And it really isn't the one that Stephen Jay Gould talked about in his book or in his essay, Darwin's Delay. It was more Darwin's not wanting to run afoul of the dominant voices in his discipline at the time with regard to human variation, the polygenists. But once Darwin had established himself as a leading figure in science, he was able to take on this question of, were the variants of humankind actually members of the same species, or whether they were different. So in The Descent of Man and Selection in Relation to Sex, which was published in 1871, Darwin recounted that different naturalists had named between 2 and 63 human races, depending upon their criteria. But he reasoned that these physical differences could not be of great importance. Otherwise, natural selection would have eliminated them a long time ago. Now, Darwin wasn't completely correct in this supposition, and in part he wasn't correct, because he didn't have a fully functional theory of heredity, which could account for how natural selection acted to change allele frequencies in populations. Now, we're jumping forward into the 20th century. By this time, the neo-Darwinian synthesis has given us a unification between Darwin's natural selection and Mendelian genetics. And now people had a much better sense of how the differences in human populations had come about. Now, it might surprise you that in July of 1950, the New York Times had a front page article entitled "No Scientific Basis for Race Bias Found by World Panel of Experts." Now, that story was reporting on the United Nations Educational, Scientific, and Cultural Organization, UNESCO 1950 Statement on Race. And this is what they said, quote, "Racial doctrine is the outcome of a fundamentally anti-rational system of thought and is in glaring conflict with the whole humanist tradition of our civilization. It sets at naught everything that UNESCO stands for and endeavors to defend." Now, prior to World War II, the Institute for International Cooperation had attempted to draft a similar document as to not offend Hitler. But in 1950, the UNESCO statement was immediately denounced by former Nazi race scientists such as Hans Gunther, Eugen Fischer, and Franz Lenz. Now, you can read a definitive history of these documents in the book Post-War Anti-Racism, the United States, UNESCO, and Race, written by Dr. Anthony Hugh Hazard. I am proud to say that he was once an undergraduate student in one of my classes. Now, the 1951 statement, however, which had 21 drafters and reviewers, none of them, again, from former Axis countries, had one historian, E. Franklin Fraser, several humanists and sociologists. The 1951 committee had 14 anthropologists and geneticists from the USA and Europe, such as Dunn, Montague, Dobzhansky, and Huxley. Now, this 1951 committee accepted the notion that humans can be arranged within biological races, but that not all human groups could be unambiguously fitted into racial classification schemes. Despite that, Fritz Lenz stated that the 1950 UNESCO document ran against the science of eugenics and that the psycho-hereditary differences in and between races were more important than the physical differences. Now, he still maintained, by the way, that Jews were a race distinguished by the former. In 1972, Richard Lewinton published his famous paper, The Apportionment of Human Diversity. Now, this actually was part of a study of a scientific question that really, again, was not driven by attempting to look at human biological variation. And this question that evolutionary geneticists were concerned with at the time was exactly how much genetic variation existed within species. The classical theory of selection that was put forward by people like Sir R.A. Fisher presented an idea that there should be very little genetic variation simply because selection would remove most genetic variants to low frequency, and only the most fit variants would be retained in the population. And this was in contrast to the balance theory of people like Thaddeus Dobzhansky that felt that there would be far more genetic variation than the classical theory could account for. Now, with the development of protein electrophoresis, people began to take measurements of populations of all sorts, many of them, for example, fruit fly species, and they were finding far more genetic variability than the classical theory could support. And so when Lewinton studies humans populations, he asks the question, how much genetic variability exists within groups that were called race versus the genetic variability between such races? And he found, surprisingly to some, that there was far more genetic variability within these groups that had been historically classified as biological races than there were between them. Now, the logical extension of that, if there's more genetic variability within groups than there are between them, then these groups are in fact not really groups. Now, Lewinton wasn't the only person who found this. Aaron Roy Kahadri and Natatashi Ney published a paper in the American Naturalist in the same year that found the same result, more genetic variability within so-called group called races as between them. And subsequent studies of human biological variation going past Lewinton have consistently found that result. Now,
So we have about 1 minute left.
wow, 1 minute. I guess my timer is absolutely wrong. So I'm quickly going to say that the neo-Darwinian synthesis came up with results that essentially completely questioned the idea that biological races exist within humans and called into question the whole notion of the subspecies. So I'm going to read you this one last quote from Ernst Mayr, who says, "The concept of subspecies is fallacious. Species are not composites of uniform subtypes, subspecies, but consist of an almost infinite number of local populations, each in turn and sexual species consisting of genetically different individuals. The difficulty of the subspecies concept are intensified if one considers the subspecies not merely as a practical device of the taxonomist, but also as a unit of evolution. The better the geographical variation of the species is known, the more difficult it becomes to delimit subspecies, and the more obvious it becomes that many such delimitations are quite arbitrary." So I thank you for your attention, and we'll go on to the next speaker.
Great. Thanks so much, Joe. We will now hear from Dr. Andrew Clark. Dr. Clark is the Jacob Gould Sherman Professor of Population Genetics. He's the Associate Director of the Cornell Center for Comparative and Population Genetics and Interim Chair of the Department of Computational Biology at Cornell University. Andy?
All right, thank you. I will launch my screen here. So I'm going to ask a somewhat more specific question about the application of population differences. I'm going to sidestep the issue of labeling these as races, but just think of the departure of the human population from being one completely mixed thing to some degree of substructure, and ask the question, now in this day of having complete genome sequences of literally millions of people, why don't we sidestep the issue of race or population structure altogether? Because all the information should be, in principle, in the genomes of the individuals that we're studying. So having any kind of label on them seems like it's less information than we already have in hand, which is their genome sequence. And the most important take-home lesson from this whole thing is that, no, in fact, it still does matter that populations, groups of individuals have some differences between them. And we're going to focus on this application of using genome sequence and genetic differences for trying to identify genes that are conferring differences in risk of complex disease, where complex disease just means diseases where there is a genetic basis, but many, many different variants are necessary. And in the early days of trying to identify differences among humans, these so-called single nucleotide polymorphisms. This is even before having the complete genome sequence. There was a sampling, and Pilar talked about this at some length, of these sort of regions of the world or population groups as were vaguely thought of by different people. So they sampled Europeans and Asians and Africans. The Africans in these early days were mostly from Nigeria, but we'll get onto that later. And there was this remarkable circularity that arose, that by identifying these populations as these discrete sort of units of sampling and then collecting the genetic information, you basically couldn't escape from this observation that was obtained, which is the genetic variation makes them look like absolutely discrete, separate entities. And this method of ordination, by the way, these so-called principal components, is just a means of graphically displaying the genetic differences between those groups. And we won't go into the details of that, but it's widely used in this scheme. So it was realized, of course, this was not by any means a sampling of the entire global population. So if we sample a bunch more populations, and this was part of the HapMap project of NHGRI, you realize when you sample African Americans, when you sample Mexicans and so forth, there's some filling in of the spaces between those three corners of the triangle that one initially had. And then, of course, today, if you look at the NOMAD project, which is a very much larger project, you end up with this sort of complete filling in, almost virtually complete filling in of this space of genotypes, where now it looks like there aren't discrete boundaries. There's still some degree of separation of the most extreme groups, but it's still a filling in between them. I will say, of course, statisticians aren't content to just sort of leave it at this. They'll keep on trying to come up with methods that tease things apart. And these methods that are widely used for single cell genomics actually have been applied to human genetic data. And here there still is some ascertainment of who should we sample in these studies, but this NOMAD project is literally an attempt to vacuum up all of the data of human genetic variation. So it's tens, hundreds of thousands of samples, and yet still there is some degree of distinct sort of population grouping if you use the right ordination methods. And these methods are exquisitely sensitive to the fact that there might be a particular allele that's found in one population and not others, or very subtle differences in allele frequencies between them can still make them look like they're completely separate entities. So that's a caveat. All right, so let's get back to the real problem that I wanted to discuss, which is this idea of using that genetic variation for identifying genes that confer differences in disease risk. And the method that's widely applied, thousands of papers have been published using this very same method, is known as genome-wide association study, or GWAS. And basically, one collects a large sample of individuals, typically a kind of a cross-section of a population. In some cases, there's a case control study where there are individuals with a disease versus individuals without a disease. But in either case, what one does is one identifies many, many single nucleotide variants, so polymorphisms in those populations. And for each and every single SNP, one at a time, you write out the three genotypes of that SNP. So one could be homozygous for one allele or the other, or heterozygote. And for those collections of individuals of each of those respective genotypes, you determine what the trait value is. So this might be, do they have the disease or not? What is their blood pressure as a quantitative measure, which is sort of the way this one looks like it's plotted. And you ask the question, is there any statistical relationship between those genotypes and the phenotype, this measured trait value here? And as you can see, the way this is plotted, it basically looks like a linear regression problem. And we're trying to determine, is there a slope for this line that goes through those points? And we're trying to test the null hypothesis. Is that slope different from zero? In other words, is there an association between this SNP and that phenotype? Now, when you do this, you find many, many SNPs that do have an association. This association is for the most part, very weak, but sometimes it does attain statistical significance, even in the face of doing those many thousands or millions of tests for a given genome sequence. All right, so if that slope is different from 0, we conclude there is an association between the SNP and the phenotype. And this association, I want to remind you, is a statistical thing. It's not saying that this SNP causes that phenotype. So this issue of causality is critical here. And this experiment alone does not infer causality. It's just inferring the statistical association. The results of these GWAS studies are then typically conveyed by the single plot, this so-called Manhattan plot, that displays the significance as a minus log 10 of the p-value on this axis. So a higher point up this axis means it's more significant, a steeper slope and more clear slope, versus all the SNPs arrayed in the genome across all 22 chromosome pairs and sometimes the sex chromosomes. And so these sort of towers indicate regions of the genome where there's a significant association. And I want to point out, often it's not just a single SNP, but many, many SNPs. And many of these SNPs probably have nothing to do with the disease, but they're simply statistically correlated, a thing we call linkage disequilibrium, with the variant that is actually important for the disease. And it's indeed possible that none of the SNPs that were actually scored have that direct causal effect, that these are all just linked to that SNP and therefore statistically are related to each other. All right, so far so good. We've talked about the fact that there seem to be some differences between population groups. We talked about these GWAS studies. What about how do we put them together? What if there are population subdivisions within a GWAS study? And the initial recognition was that, oh boy, that can cause a problem. So if there's any kind of subdivision within your sample that you're doing a GWAS in, you have a problem. So suppose a disease is common in one population and rare in another. Then any SNP that's common in the population that has the disease that's common, and on the other hand is rare in the population where the disease is rare, those will spuriously look like they're associated. There will still be a statistical association, but it clearly is just because of the population subdivision. It's not because of a true association with the disease. So that's a false positive. And the initial designers of these GWAS studies realized, ooh, we really have to avoid this problem. So we need to pay attention to these sort of population groupings, any way you want to define it, of individuals where the groups have different allele frequencies. You don't have to call them races, there's the geographically different groups with different allele frequencies will cause a problem in the GWAS approach. So several solutions have been proposed to deal with this. What do you do? So first of all, sample only from one homogeneous population. So the biggest early GWAS studies were done in the UK, the so-called Wellcome Trust Case Control Consortium was very carefully chosen from people who were born in the UK, and all 4 grandparents were from the UK, and on and on. And so it was a rather homogeneous population, not completely homogeneous. There are degrees of relationships and so forth within them, but that was one possible solution. Another was to actually include factors in the model that try to explain the differences between the populations. That is, from the SNP data, you could do that principal components analysis trick that I showed you, where you can sort of plot them. That method gives you weights of the SNPs in determining the differences between the groups that you see, and those can be included as cofactors in the statistical test for GWAS. So to the extent that there are allele frequency differences between populations, you sort of control for it with that method of principal components. This has been very widely used, even when the thought was that everybody was from one population. There's some degree of substructure within even like a strictly European sample. There's differences between different European groups, for instance. But it's not perfect, and we'll talk a little bit about the problems later. And then, of course, there's the other possibility, which is do a GWAS, a complete GWAS in each population. So don't be content to just stop with your homogeneous, quote unquote, European sample, but to repeat these studies in other groups that might be at risk. But the history has shown that, well, there was a lot more money and interest for one reason or another to do this in Europeans first. So this is a plot from Alicia Martin's paper from 2019 showing the sort of number of individual GWAS studies that were just European. This red bar, you can see it was, in the mid 90% of all the GWAS studies that have been done in Europeans. The Asians caught on, so Japanese and other Asian populations were studied, but others, Hispanic, African-American, African, and so forth, were much understudied and really are to this day. So this bias in the sampling is an important limitation of these approaches to really truly identify genetic variants for disease across the human species. So I'm going to shift gears now to talk about a little bit different problem. It'll relate to GWAS and in fact relies entirely on GWAS, but it's another application of genetic variation and relating it to disease risk that is highly impacted by population differences. And this is a thing called polygenic scoring. So it proves to be one means for doing genetic risk prediction. And for various reasons, risk prediction is likely to be of some importance in both considering therapeutics and also considering different ways of dealing with the risk of these complex diseases. For instance, changing your dietary or exercise habits or whatever. So the basic idea of polygenic scoring is you start with a very large, well-executed GWAS study. You determine which SNPs are the most likely to be causal in their association. So I sort of initially said that the GWAS doesn't really infer causality, but there are tricks for determining which SNP is the most likely to be causal out of a collection of those SNPs that are in high linkage disequilibrium. So you do some pruning of the SNPs to pick the ones that are most likely to be causal. And in the ideal world, if you knew all the SNPs that are causal to a disease, then we truly would be out of the woods and wouldn't have to consider these population considerations. On the other hand, because we're relying on these statistical correlations between SNPs, this linkage disequilibrium, these differences in frequencies between populations, that's what causes the problem that requires that we kind of somehow integrate population identities in these analyses. So then polygenic scoring finally works by using the slope of the effects of each of those individuals' genotype on the phenotype, what I showed you with the GWAS studies, and you simply add up the effects over all the SNPs. So this sum of those effects, you take the individual's genotype and sort of put those on a scoring of 0, 1, 2 for the genotypes, multiply that times the slope to get what is the predicted effect of that genotype of that individual on the phenotype, and add them up to get the polygenic score. That sounds absolutely silly that they just add up, but it actually works fairly well. And this is from Thereson's paper showing if you just rank the sample in terms of the percentile of this score, this polygenic score, and then go back and ask what's the prevalence of the disease that this GWAS was done for, you see this correlation between them. And in particular, those at the very highest risk really do have this rather augmented, often a risk ratio of as much as 5 or 6 times that of the general population.
Andy, we have about 1 minute left.
And so Alicia Martin also showed that this, as nice as this method seems, is also severely impacted by population differences. So it's very non-transferable to use a European GWAS and try to predict, for instance, the risk in African populations or East Asian populations. So again, one needs the data from the particular population to make these predictions, because the allele frequencies, the linkage disequilibrium, all the embedding of those things that you're using to associate with the risk do depend on the different populations. So I'm going to speed up a little bit here. The idea of using statistical tricks, why don't we just use linkage disequilibrium and make corrections? This could work, except for the fact, again, that you need all that information about the pattern of linkage disequilibrium within each population to get the particular polygenic scoring to work. In mixed populations, there's a particularly serious problem. So you have individuals who are admixed and have segments of their genome from different populations. And that's illustrated here. Kasia Brick had this approach for actually mapping segments of the genome of individuals of African-American ancestry. Segments of the genome are from one population or the other, and that differs widely from one individual to another. And so the take-home conclusions then, and there are 6 of them, I think. First, human populations do display differences in allele frequencies. This is just by virtue of the fact that they've been geographically isolated for long enough periods. It's a very complex picture of the sort of populating of the world, but to some extent we can reconstruct that just from these allele frequency differences. Most of those differences are quite small, as Lewinton pointed out and Joe pointed out, but in aggregate, they do allow one to see some differences even among closely related populations. There also are differences in the correlations between SNPs and then the correlations between SNPs and phenotypes. And this is the rub. This is the challenge that forces us to pay attention to population groups, even today in this age of having whole genome sequences. Ideally, we do GWASs in each population group, and GWAS and polygenic scoring can't ignore those population differences, but that someday, perhaps by being able to reconstruct segments of the genome that came from each population, we'll be able to improve these methods. And I believe it's the next section where we'll talk about sort of the future of the use of population groups in human genetics. And I'll have to end right there.
Great, thank you so much. So finally, our final speaker is Dr. Rena Bliss, who is Associate Professor of Sociology at Rutgers University. Rena?
I'm trying to start my video, but it doesn't seem to be working. Okay, wait, let's start my video. There we go.
All right, great.
All right, and I'm going to share my slides. Can everybody see that?
Yes.
Okay, great. All right, great. So thank you very much for having me. I'm really happy to be a part of this important conversation. And my research for the past 2 decades, as Pilar was saying, has focused on the ethical, legal, and social implications of genomics, especially around how it has impacted common understandings of race and ethnicity, and also how our collective concepts of human differences and genetic variation, human genetic variation, have evolved and are evolving as a result of the implementation of ethno-racial classifications in genomics. So to a social scientist such as myself, race and ethnicity are terms that we use to define ourselves, and they're aspects of our identities. So they're based on ideas about our biology, our bodies, but they are not simple proxies for human genetic variation. They're political terms, and they started out as folk ideas that expressed rather unscientific guesses about underlying biology. But they've become socially meaningful and have become real in the sense of built into our everyday lives. Like how we think of ourselves and think of other people, how we treat people, how we're treated, what schools we can get into, jobs we can get, also fields that we can realistically go into, where we live, whom we partner with, whom we marry, whom we have kids with, and as so evident in US politics, where we are safe. So I've been working in this arena to help translate what we've learned from genomics to the broader science community and to the public. And at the same time, I've been trying to keep all of us working within genomics on track, clarifying and communicating with the public about the difference between human genetic variation and socio-political stratification. The latter being the reason why we have federal classifications like the Office of Management and Budget, or OMB, or census categories. These are, within science, are targeted enrollment categories. So not to signify biology, but rather to track inequity and exclusion. So today I'm going to talk about 3 of my studies in this arena, but I want to start by giving you my conclusion, which is that all too often, genomic population descriptors have connoted racial difference, whether by mimicking racial categories or by referring to some kind of continental old world ancestry, and thereby inspiring the public to see genetic clusters in racial terms. And also by implying that the differences that actually matter to us, so disease prevalences and our life and social outcomes, are due to human genetic variation bracketed from politics. So my main argument is that we need this to end. We need to remove racial and quasi-racial continental groupings from genomic population description. And we need to use finer resolution population descriptors. And finally, we need to only be using racial terminology when we're studying racism's biological effects. So let's take a look back. So when I first got into studying the use of population descriptors in genomics, I thought that racial classification by genetics was a thing of the past. But like this cover of Scientific American from 2003 shows us, the advent of genomics did not eradicate scientific uses of race as a proxy for human genetic variation. In fact, it really just kicked up debate. So in 2000, as the Human Genome Project published its draft map, we heard a lot of mixed messages coming from genomics. Project leaders initially told the public that mapping the human genome proved that there was no such thing as race, that we were more than 99.9% the same. Race wasn't genetic, it was social, it was environmental, it was a social construct. But the following year, debate broke out, and Francis Collins and other Genome Project leaders said that we actually needed to figure out the relationship between race and human genetic variation. So there was something there, and we needed more genomic research to understand what that was. And by 2003, at the end of the Human Genome Project, when this Scientific American issue was released and the International HapMap project got going, genomic databases were being configured in quasi-racial or ONB-esque terms. So the Polymorphism Discovery Resource, the SNP Consortium, the first phase of HapMap, they were all collected, but also databased and disseminated in alignment with a continental comparative ONB-esque framework. And beyond these large-scale sequencing projects, private sector pharma and biotech databases were also shaping up in racialized terms. When I talked to scientists heading up sampling and databasing, disseminating, and in these cases, marketing at places like PearlGen, DNA Print, I also found this trend afoot there as well. So at the same time, research journals were trending towards using a continental axis of difference synonymous with ONB's sociopolitical classifications, asking researchers to use racial or quasi-racial categories in their research dissemination, so in their pubs. So it became commonplace to compare the DNA of people of European descent with those of African descent, Asian descent, et cetera, upstream and downstream of research. That's why in Race Decoded, I argued that genomics was reinforcing older 19th century notions of race as mere genetics, i.e. as, real old world continental genetic populations, as opposed to sending a really new message about race. And this was a real problem, given that everyone that I spoke with, almost everyone I spoke with, seemed to agree that pre-genomics continental or straight up racial concepts of human genetic variation of the kind of Caucasoid, Mongoloid, Negroid kind was a very poor proxy for the human genetic variation that really matters to us. And also, they agreed that there's more variation within Africa than anywhere else. So there's no such thing as an African race. So I concluded that we needed to formulate a robust gene environment science that would be co-led by genomicists sensitive to population classification issues and also social scientists sensitive to socio-political stratification. At this point, I also began conducting a study on original research reporting in Nature Genetics with my colleague, Aaron Panofsky, who is the director of UCLA's Institute for Society and Genetics. We found that there was a general trend toward using population descriptors in the first place in publication, but also quasi-racial descriptors. And also we found that researchers were shifting within their publications from very specific labels to general, continental, easy to translate into race labels. So in the space of one paper, for example, you'd see the title seeming to make some kind of race-based claim. And then in the weeds of the methods or supplemental materials, you'd see that the population under study or populations under study were defined in other ethnic or sub-ethnic terms. And talking with scientists, we found that scientists preferred ambiguous terms as opposed to standardized terms. And they did so because they were more flexible to different parts of the research process, but few were aware of the trend toward using quasi-racial descriptors. And of course, this was just fomenting the racialization that everybody was opposing. As Dr. Panofsky and I were conducting this research, I also began studying the latest in gene environment science. And at that point, there was a new field of genomics emerging called social genomics, popularized at the time in the media as genopolitics, genoeconomics, sometimes genosociology. And again, it was a really thrilling opportunity, as these fields that were, these emerging fields were based on totally new transdisciplinary partnerships between social and genetic scientists. So this was really that gene environment science I was hoping for and calling for in the conclusion of Race Decoded. And it was led by people from the social scientists who were already experts at studying the social causes of whatever phenomena they were now using genomics for. Things like educational outcomes, financial and political decision-making, sometimes violent behaviors, sexuality, and many more phenomena. So for my next book, Social by Nature, I documented this emerging field's history, and I situated it in the broader science community's study of these social phenomena using genomics, so the broader sociogenomic science that was evolving in the 2010s. And again, I really saw this triumph here, with social science and genetic science merging in very important creative new ways. But in looking at its ELSI and in particular, its population description in these early years, there I found a lot of the same issues with, genomics of the earlier 2000s. A lot of invoking racial classifications, for example, studying various forms of aggression, what reporters like to call warrior genes in minoritized populations, meanwhile studying entrepreneurialism in participants of European ancestry. Also making comparisons about things like violent delinquency and sexual promiscuity between categories such as white, Black, Hispanic, and Asian, the OMB categories, but really more generally just using the racialized databases of projects and companies. So a lot of this business as usual across genomics. And I saw almost zero analysis of how discrimination based on racism was affecting the body and translating into health disparities. And instead, more of a drive for identifying DNA variants that social policy could be tailored to. The idea being that we could genotype youth and of course adults as well to determine what kinds of educational programs or social policies they would best respond to, which I think comes from a very progressive desire to create social equality, but again, without studying the pliability of racial identities or categories, even as they were being invoked. This was more of that kind of move towards the continental comparative. So my analysis was a depiction of the early years, and many scientists have since changed course and also written about these issues in detail. But I still worry that there are many slippages and things left undefined. And so that will continue to march us towards a new, albeit well-meaning form of eugenics. So not genetic engineering via controlled breeding like old school eugenics, but social engineering based off of our DNA that could continue to be in a racialized form. And in ways that could track different groups to different futures. If we have different health insurance plans, or we have different treatment programs, or we have different medical facilities, or we have different schools. This is if we're not careful. So it may seem dystopian, but ill-defined, unscientific notions of human genomic variation, human genetic variation have already had these very effects. So I want to get back to my takeaway, which is that population descriptors that connote racial difference have been built into the infrastructure of genomics. And we see it in data collection, we see it in data storage, we see it in data dissemination. And we also see it in the wider social sphere, where comparisons of different kinds of DNA collections have shown us that DNA samples from those identified as European or white comprise the majority of databases where you opt into research or do so for recreation, while DNA samples from those identified as Black or Latinx comprise the majority of non-optional forensic databases. So we need to figure out how to end this. So I say that we start here. First of all, let's remove racial and quasi-racial labels from genomic population description. Second, let's use finer resolution population descriptors akin to what you see in the fine print of most studies. And then third, let's only use racial terminology when we are studying racism's biological effects. And yes, of course, we will proliferate categories. And yes, we will have different categories for different purposes. But we need to figure out how to disentangle OMB and eliminate the continental comparative. So I'm hoping that we can figure out better ways of using targeted enrollment today and going forward. Thank you very much.
Great. Thank you very much, Professor Bliss. So I'd like to thank all the speakers for their very rich presentations. They've laid a really useful foundation for our discussion. And now I'm also thrilled that we have so much time for discussion. Thank you for being so good with your presentation times. We will begin with questions from the committee members. However, I want to encourage our audience that has joined this webinar to submit your questions in the Slido box, which is underneath the webinar broadcast. And I promise I'll get to as many as I can. So now I'd like to ask the committee members, along with our panelists, to turn on their cameras so that we are able to see you. If you have a question, please use the hand raise function and I will recognize you. And don't forget to turn on your microphone. And Rena, can I ask you if you could stop sharing and that way we could see everyone?
Yes.
Perfect. Thank you so much. So Dorothy, you have your hand up.
Thank you. And thanks so much to the speakers for such illuminating and interesting and important comments. I really enjoyed and learned from what everyone said. I have a question for Andy, if we may use first names.
Sure.
And that is, I thought that your point was really important, that we need some kind of substructure in genomic research because people have differences between them. I think I'm almost quoting what you said, Dr. Clark. And yes, of course, the whole point of this discussion is to figure out what kinds of substructures would be useful in what kinds of research and what kinds of research questions and designs. And people have differences, people in families have genetic differences between them. So the question is, what groups? What are the groups? How should we identify these substructures? So I was struck that in your presentation, you used it seemed as biological groups, races. The only group you mentioned, populations you mentioned, were Africans, Europeans, Asians, and similar groups, which seemed to be what Dr. Bliss just mentioned as quasi-racial groups. I think they're generally identified as quasi-racial groups, although if they're not, I'd be interested to know how you selected those groups to identify. And that seemed to differ from what the other speakers were saying about the politics and history of and meaning of race, and also the role of environment, which didn't seem to be as salient in your presentation as the others. Yep. So I was just struck by that difference in the takeaway, the main points of the presentation, and wondered if you could respond to that. Again, specifically what you meant by Europeans, Africans, and Asians.
Big question. So thanks for that. So Yeah, I didn't pick the groups. I was just giving examples of the way the studies had been done. And they were mostly, from the 30,000 foot, let's just pick geographically different areas of the world where there were individuals that seemed like they had relatively little recent common ancestry. So that's the primary issue, is if they're in an area where there's a lot of mixing and intermarriages, then the allele frequencies, the variants in that group would be shared within that group, but different from those that are across the sea and in a different area. So I'm sorry that I had to use the labels that were used in those papers, but that really is the fundamental thing. But the key issue we do need to get clear, which is it's not just that there are differences between people. So imagine you have a study where you have the entire genome sequence of everybody in this, let's just say, island. We'll call them Island A and Island B, and they've been separated for a long time. Those islands, because they've been separated for a long time, the allele frequencies are different from each other. There are differences in this sort of genetic structure, the correlations between those variants in individuals. So you have those two islands. You have the whole genome sequence of individuals on this island and this island. And then you try to assess, like a GWAS study, or you try to do this polygenic scoring. The point is, if you mix them together and didn't pay attention to what island they came from, you would still be much worse off than if you had the information about which island they came from. And it doesn't have to come from up front. You can infer what island they came from the genetics. But even then, although you have the genome sequence of each individual, you need to know this individual is embedded in this population, and that establishes the correlations between the variants and perhaps the correlations between the variant and the disease. So your ability to predict disease risk of individuals, even with whole genome sequences, is improved by having knowledge of the population of origin. That's a very subtle but critical point.
No,
you cannot escape it.
Yes, I absolutely understand that. I guess the question for us is, what are those groupings that would be useful? And certainly Africans, Europeans, and Asians are not like separate island populations that haven't had anything to do with each other. Even the term Europeans, does that Europeans aren't all of the same ancestry, and of course as Dr. Blissigan mentioned, Africans have the highest amount of genetic variation. So on the continent of Africa, as I'm sure my colleague Sarah Tishkoff would point out, there are lots and lots of populations that could be identified as significant. So again, but I understand what you're saying. You were simply using the groupings that others have used and not necessarily endorsing that those are the population descriptors that would be useful in genomic research in the future.
So the problem is that there has to be some identified sampling up front that isn't necessarily based on genetics. But if you could truly do that geographic grid sampling, you would still identify clusters by these various methods that combine similar genotypes. Like that picture that I showed from the nomad data that showed how distinct the Ashkenazi and the Finns were. That sort of grouping comes out from the data. It's not all driven just by the sampling. And so in a way, that's the most useful kind of grouping to think about, is whatever forms a cohesive group in these various ordination methods.
Okay, I'll let others ask questions.
Great. No, thank you, Dorothy. Actually, I wanted, before I go to Anne, I'm just noting Pilar's comment in the chat, and I just wanted to give Pilar, you an opportunity, since this is related to this question about population and how do we define it and which context. So if you wanted to jump in at this point.
Well, thank you. Yeah, my question was also for Andy, and it was a little bit similar, I think, that I was just wondering, we've had all these reports of polygenic risk scores not generalizing. And a lot of times people talk about that with respect to racial groups. I am wondering when people talk about, well, we would need to repeat these PRS studies in every population, which is a comment I have seen, like, what would that mean, right? Does that mean that we need to do it in Finns and people of Ashkenazi Jewish ancestry, as well as, what does it actually, how do we understand a population in the context of PRS? And what do you think explains the lack of generalizability in that context?
Yeah. No, it's a great question. And you actually made a really good point about the environments being different as well. And that actually is a key aspect as well, that the sort of opportunities for different genotype by environment interactions across populations also is a key thing that probably does erode the quality of polygenic score when you try to transfer it to another population. But in essence, you have to realize polygenic scoring is based on these statistical kind of correlations between SNPs and between SNPs and the phenotype. And so do we need to do a GWAS in Finns? Maybe not, but if we do it in a closely related population, so if we do it in Swedes or something like that, we'll get a better result than if we do it just with English or Americans of European ancestry, or whatever you want to call it. But the key point is the populations have to be similar in that structure. And you can tell that quantitatively from these ordination methods, UMAP and polygenics or principal components analysis. But there is the practical problem that we can't afford to do it in every single small population. But if you are descended from a population on an island like Tonga, where there isn't any other closely related population, it's a small population with many different allele frequency differences, you're kind of out of luck with respect to having an accurate GWAS available for you, to the extent that having a polygenic score helps you, which is another debate.
Right.
Yeah.
Great, thank you.
Great question.
Dr. Morning, yeah.
Sure. Thank you. And I'd like to start off by thanking all of the panelists for these really excellent presentations. I have one question for Pilar Osorio and one question for Andrew Clark. And I think this dovetails on what we were just talking about. So Dr. Osorio, you spoke about grid sampling very briefly, and it just came up again now. Could you just say a little bit more about the fate of that idea, why it hasn't gotten more traction, and maybe your thoughts about the promise it might hold? And then my question for Andrew Clark, I guess it's really clarification. When you brought up the challenge, the problems of false positives, of SNPs that are false positives, I just want to understand that is a problem that will present itself even in a population that we might consider a fairly homogeneous one, no? So I guess I'm just interested if there are any measures of how much the introduction or the consideration of population structure in GWAS helps prune back or reduce the problem, the degree of false positives that one might have to contend with. So I'd just be interested to know if there's any kind of measure or indicator of how much population structure incorporation helps relieve the problem of false positives. Thank you.
I could go first. And, I'm not sure how satisfying my answer will be to the question of grid sampling and why it really never had much of an impact. I think, first of all, few serious genome scientists have proposed it, but it was something that came up, for instance, in the 1000 Genome Project. And, I would only be guessing at what's going on in other people's heads for why this really was never, it never got much traction. But, and my feeling is because genome scientists often do have beliefs about the importance of the kind of, especially the continent level population history, so to speak. And so felt like it would either be much more technically complicated to analyze data collected through grid sampling. I mean, I think nowadays, because we have PCA analysis well developed and things like that, people might not be as worried about the difficulty of analyzing data that way that was collected through grid sampling. But I think there was a feeling that this would introduce a level of complexity that that might make it hard to analyze. And, again, that reflects some preconceived ideas about the nature of the endeavor of doing genome science, right? Yeah.
I'll just say there is grid searching in some instances. So in the Framingham study, for instance, there was some effort to sample. So on a very different scale, not global scale, but at smaller scales, there's citywide searches, particularly for things like in epidemiology of infectious disease. Those are often sampled by grid search. And so it does have some application. But to answer your question, Anne, the Wellcome Trust Case Control Consortium, that was all UK, whites from UK. And if you totally ignore and assume that it's completely homogeneous, you don't do quite as well as you do if you try to accommodate something a little bit different from thinking of them as being in subdivided populations, but rather differing in their degree of relatedness one to another. And so another technical way to deal with the fact that it's not just this homogeneous, equally kind of distant individuals from a population is to include in those models a weighting by what's called a genetic relationship matrix or relatedness matrix. And so you can actually calculate from all the SNPs, what's the sort of degree of similarity. And for instance, if there's a pair of siblings in the sample, you can see that if they share a lot of their genome, 50% in expectation, and if they also share a particular phenotype, you're giving an unusual weight to those individuals compared to others in the sample. So you need to sort of weight them by their relatedness. And that works quite well. And that's used in essentially all GWAS studies now. I can't give you a quantitative number on how often, is a SNP that you think is really important and a true causal SNP or whatever missed if you don't have this weighting, or how many more false positives do you get in the absence. But it definitely cleans things up. It is an improvement to consider those factors, even in a sample, as you suggest, that you believe to be from one homogeneous population. Yeah.
Thank you both so much.
Great. Dr. Graves.
Yeah, I mean, this is more a comment than a question. But one of the things that not everybody in the audience is familiar with is when we talk about GWAS and PGS, exactly what percentage of the variation in the phenotype, particularly complex phenotypes, that these methods are actually predicting. And so if you start jumping up and down about, wow, you've got X number of SNPs and you're predicting this amount of variation, but the variation is 5% in the phenotype in question, that means that 95% of the variation in phenotype is not predicted by the SNPs. And so we need to be really careful about the way people report GWAS studies. I do GWAS myself, but I do it in systems in which I can rigorously and assiduously control the environment. And I can rigorously and assiduously control selection. And I can use techniques statistically to eliminate, SNPs that really aren't causing the phenotype in ways that people who are doing research as humans just can't do. So we really need to be careful about, people jumping up and down about what human GWAS studies are actually predicting.
No, I completely agree, Joe.
Okay, great. Aravinda?
Yeah, a comment and then a question actually for all four speakers. By the way, thank you very much for using your 15 minutes to provide a very cogent view of both history and, at least for some of you, where we might go. I'm actually very interested, just like all of you, to try and articulate over the next however many months how to move forward, or at least know whether we can move forward. So there's actually no doubt that the past, I know people say the past is prologue, but I think I want to move through this prologue. There are many, many problems, and I think some we should talk about explicitly, maybe not today, but remember that there are problems. The history of the use of genetics has been differentially painful and of use to different people. There's actually no doubt. When I walk the streets, nobody's asking me where I've come from. They made up their minds long, long time ago, and I'm at the better end of the spectrum. And I think all geneticists have to really have to remember this. The second is that I think some of you mentioned is geneticists particularly, they may be other scientists, but I don't read the literature perhaps just as well in other non-genetic sciences. But clearly in the molecular sciences, we use labels reflexively with very, very little thought. And I think this perpetuation, which by the way, I'm going to pass some of the blames to editors of journals and professional societies. There's a lot of people who could have made this change, but honestly, we haven't. The third is, I personally, I'm actually very interested in seeing how we can improve the science. I don't think we should resort to statistical ways of doing GWAS if the idea is to find the underlying basis of whatever phenotype. I don't think we should stick to this. We should figure out, if we are interested in the molecular basis of some disorder, and by the way, the focus on disorders is in fact of great social value, which is why social categories of race and ethnicity have been used, then we should focus that that's the problem to focus. Just saying it is hard and using these labels and using the past history in the way is not doing any of us any good. Okay, we are doing GWASs that are 10 times bigger, perhaps even 100 times bigger, but they will have exactly the same problem. And so that's one of these things that I believe we can tell NIH. The other is, even for, I mentioned this internally, even for so-called single gene disorders like cystic fibrosis, it is quite clear that human diseases for sure and many phenotypes have very strong social, cultural, environmental factors. One of my mentors used to constantly tell me, he said, we are geneticists, not hereditarians, but geneticists reflexively are hereditarians. We have been. That's what the historical literature suggests. So there is a paper, there are a number of such papers. I just passed one around to my colleagues last week that shows in this particular study that atopic dermatitis is that there are differences by, in quote, race, but they are not recapitulated by any genetic differences. So I think we have to do studies that are much richer, and there are many ways of doing such studies. So what I wanted to hear from the four speakers is, if you had to leave, we have your lectures, we're going to go back through it. You have to leave, each one of you, with one lesson that this committee should really think of and emphasize. What might it be? And I am, your cat looks very much like mine, Pilar. So I'm going to start with you.
Thank you, Aravinda. Yeah, so my lesson, I tried to like quickly put it in there at the end, which is, I don't think we should fool ourselves into thinking that better sort of methods of collecting race and ethnicity information will actually improve the science. But instead, I do think that better methods and more dedication to collecting the social context data might actually improve the science and teach us really interesting things. And I do think, I mean, it will be technically difficult in some cases to, new methods, probably statistically and otherwise, will need to be developed. to analyze gene by environment interactions in intelligent ways. But I do actually have faith in the genomics community and the scientific community that there are tons of really smart people out there who could do that.
Thank you. Joe.
Yeah, so my take-home message was cut off in the slides I didn't get to, but basically it's rather simple, that our social conceptions of race and ethnicity do not match the underlying biological and genetic variation within our species, and we should never confuse the things that were created for the purposes of oppressing people with the nature of that biological and genetic variation.
Great. Andy, you want to go next?
Sure. So first of all, Aravind, I do agree that ultimately we do want to understand the molecular basis for disease. But I guess my take-home message would be, in the meantime, when we're still in this context of using these sort of population survey type approaches to identify potential variants of interest, GWAS-like methods, that even in the face of having whole genome sequence, the context of the environment and population do matter. You can't rely just on each individual's genome sequence. You need that context as well.
Okay, and Rena.
Yeah, so eliminate any chance that there is going to be some kind of continental comparative. So eliminate any quasi-racial classification, any of that, from especially when we get to the publication, research, dissemination stage of our jobs, right? So, and I think that as much as we're talking about this in terms of how to make science better and make the science better, we have to remember that these ideas and the headlines that come from the research that's done in this community impacts how law enforcement do their jobs, how professors teach, so I'm referring to actual research that's, other members of this group here present right now, right? So, and also how healthcare providers do their jobs. So it's really like, this is about how this research impacts our whole entire society in all of its different institutional domains. And it's like, it means whether people live or die. So it's important for us to realize that comparative, that thing of saying that there are these different old world populations that are like the original groups and that they've never, met and they've never mated with each other. And that, that's very strong in the United States and in other countries as well.
Great. All right. Louisa, you have a question.
Thank you. I would like to thank the speaker for all the insightful comments. And I would like to ask a question to Dr. Bliss, actually. At the end of your talk, you say that we should only use the OMB categories when we try to capture racism. Sadly, we live in a country that racism is a stratification system that keep race and ethnicity alive and generate all these racial, ethnic difference and outcome that we observe. So how do we capture that in an EWAS study, for example, if we don't have any descriptor that associate that socio-exposition, if we wish to call it, that represent race and ethnicity in a lifelong cumulative exposure or an individual?
Yeah, so I think that's the only case where you would have racial classifications, where you would sample by race and you would use OMB categories, or you would use, when you're actually measuring, for example, structural racism, epigenetic effects, that kind of thing, you would want to use classifications in that particular case, right? But if you are not doing that, which is what the majority of the research that we're referring to right now in this, it's few and far between a project that actually looks at racism in terms of its, biological effects. And so within the space of genomics, right? We're talking about population descriptors in genomics. So in that case, don't use categories that can be misconstrued as proving there to be some kind of, again, human genetic variation bracketed from politics, bracketed from the social, the social structures that are conditioning us, right?
So we
Thank
have,
you.
thank you. So we have about 10 minutes, and I do want to go to the questions that have been submitted by the audience. And notably, there is tremendous overlap with the questions that have already surfaced thus far. But I did want to push just on that last question that Louisa mentioned. Nancy Krieger has asked for recommendations for population-based research, which relies on OMB racialized categories for race and ethnicity, that we use. When we use those, we are using census-derived denominators to calculate things like population rates for morbidity, mortality, hospitalization rates, et cetera. And she is asking, should we require the use of metrics that refer to structural racism and other structural determinants, and not just focus on categories used for racialized groups? And that, I think, builds on that last discussion. And I'm wondering if any of the panelists have more to say on that.
I actually have a lot to say on that, but I was hoping that somebody else would say something first. And the one thing that is absolutely clear, and I know this committee doesn't want have conclusions, but I'm going to say this since I'm not on the committee, I can get away with it. But structural racism kills people and has been killing people from the very beginning of this society. And of course, we now know that there are levels of change that occur within individuals that can be measured. Things like methylation, for example, as indicators of biomarkers of stress. And so we need to start measuring those things because they are contributing to lifelong morbidity and early mortality, as has been repeatedly shown for a variety of racially subordinated groups, not just in the United States, but around the world. So I completely agree. We need to be measuring this because if you can't change something until you are willing to face it, and you can't face it until you measure it.
Yeah, I would just completely agree with that as well. And I think we need to get better at measuring both the structure and the biological impact of the structural racism, right? Those are two separate things, and we need to be measuring both of them. And there needs to be a commitment, right? There needs to be a commitment within the scientific community to being able to work with those data, and a commitment among funders to actually make sure that people are gathering those data. When people are within the scientific community peer reviewing papers, asking about those kinds of data, and if they're lacking, why are they lacking, and things like that.
Okay, we have another question here. A lot of the conversation and considerations on this topic are very US-centric, and the impact that race ethnicity descriptors have had socioeconomically in the United States. How do we keep this in consideration but go beyond US sociopolitical history to come up with population descriptors that are applicable and useful beyond the United States to the rest of the world? And this is in the context of genomic studies, this question. Thoughts on that?
Like I said, I don't actually think we should necessarily try to do that because population descriptors, unless you're just using how people's actual genome data fall out on a PCA plot, those descriptors inevitably partake of a particular social and historical context. And, if we do try to come up with, more generalizable descriptors, we are fooling ourselves into thinking that we are actually collecting data that are some way comparable when I think they are not.
And I agree with Pilar, because these things are very specific to historical cultural context. I can remember reading a story, a study, not a story, about who gets sickest the most and who dies the most in Scotland. And they found that working class people there were the ones who got the sickest and who died the soonest. So genomically, these individuals aren't really any different from people who are not working class, but the social subordination had impacts on how long they lived and how well they lived, and that's true all over the world. So each one of these studies is going to be context specific.
And if I could just add something, thinking about, so China has invested a lot in doing genome research. So they have over 50, quote, recognized ethnicities within China. That is reflected in some of their data. Although, like us, my guess is that a lot of their data come from like Northern Han Chinese, as they would classify people, right? But it is unlikely that studies done in the US are going to capture that level of context, ethnicity, history. And on the other hand, it's ridiculous for us to like classify Asian as one, sort of group of people. And, I think the questioner is kind of pointing out to us that where you are and who you are doing the science influences your understanding of what groups matter, right? And what kinds of questions you'll be asking. And I think, we also ought to not make it seem as though genome scientists are somehow like this unusual group of people. No, genome scientists are embedded in a cultural and social context as well. And so US scientists are doing our studies using categories that make sense to us. And part of improving this is to get people to recognize when they are doing that, right? But that we all do it.
Okay, we only have a few minutes left, and there is one question. If you were given a wish, what tool would you ask for that would greatly benefit the understanding of genetic variability and race? So thinking forward, what is it that we need in order to do a better job in terms of understanding that relationship, if indeed there is a relationship? Joe, you and
Yeah, I have an answer, but I'm always trying to give everybody an opportunity to weigh in. But I think the thing that we need the most with regard to trying to understand the phenotype, because again, the question of whether we're hereditarians or whether we're geneticists is important. Phenotypes are produced by an interaction always between genetic background, environment, and chance events. And those interactions are complex. And we've spent a great deal of money to sequence a whole bunch of genomes, but we haven't ever done it in context, particularly with regard to questions about, health disparities. So what we need is more sociological data that can more finely get a handle on how the environments of these groups are different. And of course, what would be the best experiment would be able to equalize aspects of the environment. But we can't do that with people. We do it with fruit flies. And it's amazing how easy it is to distinguish genetic versus environmental effects when you can equalize environments.
I think that a different way of using targeted enrollment and some kind of language that can require people to use those categories for enrollment if that's if there's a reason to use multiple categories, and to explain why those categories can't migrate throughout the data analysis and dissemination process.
Any other last thoughts from our panelists before we close?
I just wanted to note that there is a question from Lance in the chat for Andy. Would be a really great last thought to answer that.
Yeah, thanks. I was just starting to type the answer to Lance if we didn't have time, but yeah, so he asks about, so the measures of relatedness, can they be applied sort of to global samples, or does it only work in the context of relatively constrained studies? And then what's the relationship between measures of relatedness and population labels? What do you get from relatedness that you don't get from population labels? And relatedness measures, of course, are based purely on the genetics. So with respect to Joe's comments, which I agree with completely, that there's no aspect of context or environment or anything like that. It's just the SNPs, the gene sequences, and it's asking what fraction of the genome is shared between pairs of individuals. And of course, individuals from opposite sides of the earth do have a non-zero fraction of their genome that's shared. And this is recognized by collections of SNPs that happen to line up, these sort of identity by descent tracks, which you can see all over the world. But the degree of sharing from individuals from very disparate parts of the world, disparate populations, will be much, much lower than within a population. So absolutely, relatedness could be applied anywhere. It's much, much finer grained than population labels. In principle, you know, you could, it seems like, getting back to the point that I keep on making again and again, well, then why not do away with population labels altogether? And the population labels let you accommodate this sort of aspect of the correlation structure among the SNPs, which again, the relatedness measures do not. So you still need the population labels, even with that fine-grained, exquisite detail of degree of sharing you get from relatedness measures. Yeah, it's a puzzle. I guess the magic thing I would wish for is everybody should have a course in linear algebra and high dimension statistics. I mean, it's a very complicated problem. Yeah.
Okay. All right. So I want to thank all our speakers for a great first session. And I think many of the points that have been made will be taken up in our next session, which is focused on the future use of descriptors. We will break for 30 minutes, a little shy of 30 minutes. Please come back at 1:30 Eastern time, and we will start our next session. Thanks, everyone.